We are talking about a marine worm. This worm lives in Sea of Japan, feeds on organic particles in mud and sand regions or habitats. Here, the light intensity is acting as a stimulus. The scientists selected 11 animals living in mud and in sand, and the time in seconds before to extend their bodies from their burrows was then recorded. Okay, so here we are asked to state a suitable null hypothesis for this investigation. The suitable null hypothesis would be the mean time taken before extension in mud and sand is the same. There is no significant difference between the mean time before extension in mud and sand habitat. B.I. Draw a suitable table to display data and your calculated means. So here we will draw a table like this, and we will calculate the mean for each habitat. And this is it for BI, 3BI. 3B2, we will bar graphs, as we have two different habitats to compare. Here is the graph. We will take the least value for time and the max value for time and use it to draw our SD bars. And we are done with B2, 3B2. 3C. The formula is given, we just need to input the values. So this NA is the number of samples for each habitat, and how many samples did the scientist collect? 11, right? 11 samples. So we will write here 11 for each habitat, and the average values for each habitat. Here I have taken A for mud. A representing the mud value and B representing the sand value. And here values here are given in 3 significant figure. It's a hint that I also have to keep it's better to keep my calculated answer in 3 significant figures too. 3C2. According to our syllabus, we have to take this value of 0.05. We will calculate the degree of freedom, 11 minus 1 plus 11 minus 1, and it gives us 20. Our critical value is 2.09. Okay, explain the conclusion that can be drawn from this investigation. Critical value is 2.09. Calculated value is less than the critical value. Therefore, accept the null hypothesis. There is no significant difference between the extension time for mud and sand animals. SD bars overlap, as we can see in the graph. Therefore, data may not be valid. 3D. This animal has light sensitive pigments called opsins. Okay, these are pigments. Suggest how opsins allow the animals to respond in its investigation. Cis retinal in opsin is converted or changes into transretinal. we will basically just describe the conversion of cis-retinal into transretinal and as per our syllabus and notes, and this causes an action potential in sensory nerve, hence causing a reflex response that causes muscle contraction in the lenses. Quick is that we have circular and radial muscles in the lenses. Therefore, an action potential causes the contraction of these muscles. And we are done with question number 3.